So before I start, when is it that the DNA will replicate in a cell? When DNA will replicate in a cell? Andrea?
S phase of interphase.
Okay, very good. S phase of interphase. Okay. So during S phase of interphase is when the DNA will replicate to prepare the cell before the cell go into mitosis, where the nucleus have to divide. And then after that, the cytoplasm will divide. Okay, so exactly what is needed during DNA replication is like, imagine you are trying to make a cake. So what are the ingredients that you need to make the cake? Okay, so DNA replication requires, number one, the DNA template itself. Number two, of course, the free DNA nucleotides that you need to add one by one in. Okay, and then of course we need enzymes, because all biochemical reactions in the cell are catalyzed by enzymes. So we need enzymes. So here we need a few enzymes. So the DNA template that we talk about is the original double-stranded DNA molecule that we have in the nucleus. So each strand of the double helix will act as a template. So here what you can see is you have this two polynucleotide strands that are joined by hydrogen bond between the complementary bases. So there are complementary base pairing that hold these two polynucleotide strands together. So each of the strand will act as a template. Okay. So this will act as DNA template 1, and this will act as a DNA template 2. So then we also need free nucleotides. So how do they look like? So we need 4 different free nucleotides, which is called the dNTPs. Okay, dNTPs. So dNTPs is a short form which stands for deoxynucleoside triphosphate. So dNTP. Okay, so this is an example of a dNTP molecule. So you can see that deoxy, because the sugar is a deoxyribose sugar. So you see carbon number 2, there's no O to carbon number 2. Okay, so that's why it's deoxy. Nucleoside triphosphate. So what is nucleoside? Okay, nucleoside is the sugar and the nitrogenous base component of the nucleotide. Okay, so I will show you with the diagram afterwards here on the right side. Now remember this drawing here. So sugar, phosphate, and nitrogenous base, this whole structure is considered as one nucleotide. Okay, so one nucleotide. Now where is the nucleoside side? Okay, nucleoside is the sugar and phosphate. So when you say nucleoside, you're considering the subunit sugar plus the sorry, not phosphate, sugar and the nitrogenous base. So this is the nucleoside. So how to remember? Very easy. Nucleoside contain the base that point to the side. Okay, the base that points to the side. So it's nucleoside. So sugar and base. Nucleotide means you got all the 3 are joined together tightly. So nucleotide. Okay, sugar, phosphate, and nitrogenous base. Okay. So therefore, deoxynucleoside triphosphate. Okay, deoxynucleoside triphosphate is the free nucleotide structure. Now, of course, the base have got 4 types of bases for DNA, right? So in this case, it's adenine. So therefore, this free nucleotide is called DATP. Okay, because the nucleoside is made out of the base adenine. So the deoxyadenosine triphosphate, deoxyadenosine triphosphate. You don't have to memorize this, okay? You just have to know DATP, okay? So this is DGTP because the base is guanine. This is DCTP because the base is cytosine, and this is DTTP because the base is thymine. Okay, so after having seen that, are you familiar with ATP? So if DATP is deoxyadenosine triphosphate, what is ATP then? Chloe.
Adenosine triphosphate.
Adenosine triphosphate, very good. Okay, so where can you find this ATP? DNA or RNA? Gan Yuhan.
RNA,
RNA, why?
because ribosomes is it?
You see, when you put a D there is deoxy, right? Deoxy means you are referring to the sugar that has no oxygen there in carbon number 2. That's why it's called deoxyribose sugar. So ATP doesn't have a D in front, correct? So that means ATP, the sugar is not a deoxy sugar. So if it's not a deoxyribose, that means it must be a ribose, right? So if it's a ribose, that means this ATP can, ATP is the free nucleotide for RNA, right? DATP is free nucleotide for DNA. Okay, so this is how you can work it out. So therefore, ATP is also a nucleotide. Okay, it's a nucleotide with 3-phosphate, but the sugar is a ribose. So it's a nucleotide that belongs to RNA. Okay. So the free DNA nucleotides is a deoxynucleoside triphosphate, so DNTP, N is a general term to represent all the nitrogenous base, okay? And it has got 2 extra phosphate group. So normal nucleotide is only 1 phosphate, but for the free nucleotide, it contains 3 phosphate. So there are 2 additional phosphate there. So why do we need the additional phosphate? Is because during DNA replication, we will remove the 2 extra phosphate to release energy in order to join the 2 nucleotides together, okay, by forming phosphodiester bond. So it's very similar to when we take ATP, right? ATP, when we need energy, we hydrolyze ATP, right? We break down ATP to form ADP plus PI, isn't it? Okay, PI, which can further, ADP can be broken down to AMP, okay, plus another PI, right? So when we need energy, we break down ATP. So during DNA replication, they uses DNTPs. DNTPs. Where's my DNTPs? DNTPs. Okay. So therefore, they will actually hydrolyze it so that it becomes the nucleotide plus 2 phosphate PI. Okay. And then plus the energy that's needed to join the 2 nucleotides together. So it's very similar to the hydrolysis of ATP in order to release the energy. That's when the 2 extra phosphate will be removed. Okay, so that's for the free DNA nucleotide. So now let's look at another enzyme, helicase. Not another enzyme, the first enzyme. First enzyme, helicase. We need enzyme. So helicase, right, this name is very interesting. This name of the enzyme is like helicopter, right? Helicopter. Okay, what does a helicopter do? No, of course cannot. Of course, it flies. Now, helicopter, right? In order to fly, on top, it has to rotate, isn't it? It has to rotate. So, helicase, right? ACE is an enzyme that will cause the DNA double helix to unwind. Okay, so it's turning, right? So it causes it to unwind plus unzip. Okay, so originally your DNA is a DNA double helix. That means the two polynucleotide strand, the double strand, twist and turn into helical shape. So in order for DNA replication to occur, we must first unwind it first. So we unwind it into straight chain, okay? And then we must unzip it. Unzip is like, imagine these two strands are, are joined together by hydrogen bond. So it's like the zip that you have in your pants or your clothing. And then when you want to unzip it, means you break the hydrogen bonds that join these two strands together. So like here. So in this one here, you can see that this beginning part of the DNA double helix has already unzipped. So therefore, the hydrogen bonds between the two strands are already broken. So therefore, these two strands are now, the bases are already exposed and they are no longer joined together. So bases, so hydrogen bonds are broken when unzipping happen, unzip. Where's my unzip here? Okay. So when the two strands unzip, it means that the hydrogen bonds are broken. So therefore the bases are exposed. So they are no longer pairing. So they're exposed so that we can add in the new nucleotide by complementary base pairing. Okay. So helicase will catalyze the process where DNA will unwind and unzip. Okay. And then where the hydrogen bonds will be broken between the complementary bases to form two straight chains. So these two straight chains here is actually called the replication fork. So this region of the DNA is called the replication fork because the bases are already exposed. So therefore, replication can already occur here. That means you can add in new nucleotides already. So therefore, this region of the DNA is called the replication fork because this is where replication can start already. You can make the new polynucleotide strands already. Okay, because the bases are exposed for complementary bases to come in. Okay. So, and this process needs ATP. So unwind and unzipping requires ATP. Yeah? Okay. So the direction of DNA replication. So because the unwind and unzipping starts from here, so the new nucleotide will be added in here first. So as a new nucleotide is added in, your helicase will continue to move forward to continue to unzip the DNA double helix. So therefore, the nucleotide will continue to be added in this direction. So therefore, this is the direction of DNA replication. Okay. So this one is another diagram to show you that the helicase will unzip the DNA double helix. So you can see this blue color line is the hydrogen bonds. So in this, for these two chains, the blue color lines are no longer there. So they are already broken. Hydrogen bonds are broken. Okay. So the next enzyme that we want to look at is the DNA polymerase. It's a very important enzyme for this process. So enzyme DNA polymerase. So DNA polymerase is an enzyme that produces the polymer called DNA. Right? So DNA polymerase is an enzyme that will produce the polymer called DNA. So what does it do actually? You see, if this is the DNA polymerase, it has got two substrates that have to stick to the active site. Okay, so this is the DNA template strand here. This is the DNA template strand. Okay, these are the new nucleotides that are added in by this enzyme. Okay, so therefore now this enzyme is going to add another free DNA nucleotide, which is in the form of dNTP, to the last nucleotide. So it wants to join the free nucleotide to the last nucleotide of this new polynucleotide strand. So therefore, there are terms that you need to learn up to describe. So the last nucleotide in the chain is called the terminal. Okay, the terminal nucleotide. So the last nucleotide is called the terminal nucleotide, which is this one here. Okay, this chain is the new polynucleotide strand. So the growing, quite a growing polynucleotide strand because it's still growing. Okay, so the DNA polymerase will have active site for two substrates because two of them need to react. So two of them must sit into the active site. So the terminal nucleotide will sit at one side of the active site, and the free DNA nucleotide, so this is called the free DNA nucleotide. So in exam time, you can also use this term, free DNA nucleotide, okay? Or if you can remember, you can say DNTP. So DNTP has got a sugar, 3-phosphate, and 1 nitrogenous base. So it will sit in very nicely to the active site of this enzyme, okay? So now you can see that what happened is that this is where condensation will happen. Okay, where you will remove the condensation means you remove the water. Condensation means water come out. So you remove the water. Okay, so therefore the location where condensation occurs, they must be facing each other, right? So this phosphate has got an OH attached to it. So this OH and the OH in carbon number 3 will be facing each other so that water can be removed, which is HHO. Okay, so water can be removed. So then the phosphate attached to the O attached to the carbon. So to form this new covalent bond, okay, to join these two nucleotides together. So therefore, can you see that enzymes are being very specific because of the specificity of the active site? So on top here, it must hold the terminal nucleotide, and the bottom here, it must hold the DNTP. So therefore, the DNTP is always added to the terminal nucleotide so that the polynucleotide chain will always grow in this direction because the new polynucleotide will always be added in here, okay? And here, here, here. So the chain will elongate in this direction. It can never grow in that direction because of the specificity of enzyme, how it holds the terminal nucleotide and the free DNA nucleotide. They must join in this manner. So the next one must join here, right? And the next nucleotide must join here. So can you see that the new chain always grow in this direction? And this direction is considered as the 5'. Can you see that? Okay, the sugar phosphate backbone, the first carbon here is 5'. Carbon number 5 is 5'. And the last carbon here along the sugar phosphate backbone is carbon number 3, right? 1 here, 2 here, and 3 here. So therefore, this is 3, yeah? So therefore, all polynucleotide strand will grow from 5' to 3' direction because of the enzyme specificity, okay? So that's why when we label the direction of the polynucleotide strand, we always label it as 5' to 3' direction, okay? So therefore, this is 5' to 3' direction. This one, 5' to 3' direction is opposite, okay? So once the new nucleotide is being added in by complementary base pairing, so there will be new sugar phosphate linkage form. So this is the new sugar phosphate. What sugar phosphate? Sugar phosphate backbone has got a new linkage form. So what is the name of this bond? Lim Jing-er.
Phosphodiester bond.
Wow, very good. Phosphodiester bond. Okay, so the new linkage, is called the phosphodiester bond that join the two nucleotides together. Okay, and there will also be new hydrogen bonds. So there are two new bonds that are formed, right? First is the bond that are found in the sugar phosphate backbone, right? So there's a phosphodiester bond. Second is the hydrogen bonds that are formed between the complementary bases. So these are 3 hydrogen bonds, right? So therefore, 3 hydrogen bonds is between which 2 bases? Daniel?
Teacher, could you repeat? I'm not sure.
You're not sure that I open to the floor already, huh? Okay, open to the floor. Cytosine and guanine. Yes. Okay, who write first? Lo Xi look right first. Cytosine and guanine. So three hydrogen bonds always between cytosine and guanine. Cytosine is a pyrimidine, so guanine is here. Okay. So you need to remember, if it's 3 hydrogen bond, it's always G and C. 2 hydrogen bonds, a T for 2 is always A and T. So T is the pyramid, right? Thymine. So this is A. So even like this also, you can actually work out what are the bases there. And this is another very popular question in MCQ. Another popular question in MCQ is this one, okay? So who got a point? Lo Xi, is it? Congratulations. Lo Xi, right? That's 2 marks. Okay, so, right, so 2 new bonds are formed already when you join 1 nucleotide, and always grow from 5' to 3' direction. Now, so why always grow from 5' to 3'? Now you know already, because of the enzyme specificity. Enzyme specificity, that means the way the enzyme hold the 2 substrates. You only allow the chain to grow in 1 direction, which is 5' to 3'. Okay. So new chain always grows from 5' to 3' direction. All right. So the third enzyme that you're going to learn is DNA ligase. So DNA ligase, I will look at this enzyme like a glue. All right. So this is something like a glue. Okay. Why like a glue? Because there is a G. Okay. There is a L. GL, there's no U, there's a E. So it's a glue. Okay. So that's how I remember the function of this enzyme. If you can find any other interesting way to remember, you can do that. Okay. So to me, DNA ligase is a glue. That's it. Okay. So what does it do? So can you see here, this navy blue color is the DNA template strand. Okay. And the lighter blue or the sky blue are the new polynucleotide strands that are formed. So here I've got two fragments of new polynucleotide strands. They are not joined together yet, okay? So therefore, there's a gap here. So there is a gap here. So these are the 2 polynucleotide strands or fragments that are not joined yet. So it requires an enzyme to join them together. Okay, so this enzyme is DNA ligase. So that's why I say it's like a glue. It glues DNA fragments together. Okay. So if we glue DNA fragment together, what bond is being formed? Sarah?
Phosphodiester bond.
Very good. Phosphodiester bond. You guess one or you know one?
Okay.
Good guess, huh? Okay, good. You see, correct. Because if you look at this chain, right? Now, this is the sugar phosphate backbone, right? Along this line is definitely sugar phosphate backbone. The one that point to the center is the base, right? So therefore, the entire chain here, this line here, they are all sugar phosphate backbone. So you want to join a sugar phosphate backbone of this chain to the sugar phosphate backbone of this chain. That means you need to join the sugar to phosphate, right? So therefore, the only bond is phosphodiester bond. Okay, good guess. So DNA ligase function is to join 2 DNA fragments together. Okay, so these 2 are the 2 DNA fragments. So after you join them, you form phosphodiester bond. So in order to link the sugar phosphate backbones of these two DNA fragments together. Okay. I've got another question which I can ask you. Okay, never mind. I don't want to ask. So this is a DNA template. All right. So let's recap what are the ingredients that you need to make new DNA molecules, okay? So number one, you need the DNA template. Number two, free DNA nucleotides. It's better to write free DNA nucleotides because we also got another version of nucleotide, which is RNA nucleotides. Okay, so therefore it's actually better to write free DNA nucleotides. I'll change the notes also. Number three, enzymes. So the enzymes that we need are number one, helicase. Helicase is to unwind and unzip. That's why helicopter, turn, right? So unturn, okay? Helicase. But you must remember to write unzip. If you don't write unzip, you don't get marks. Must unwind and unzip. DNA polymerase is to add the free DNA nucleotides to the template to form the new polynucleotide strand using the template. Okay. So DNA ligase is like a glue, so it will join two DNA fragments together. So now we want to look at the entire process of DNA replication. So we can divide the entire process of DNA replication into a few steps. Step one is DNA unzips. Step two, DNA replicate, where you produce a new chain, right? The new polynucleotide strand. Then, so because there are two templates, so in these two templates, a different process will happen. So we give different name. So in one of the template, the DNA replication is considered as continuous replication. In another template is considered as discontinuous replication. So even though on both templates, polynucleotide strands are produced, but the direction they're being produced is different. So therefore it's considered as continuous replication or discontinuous replication. So let's look at it now. So we have got, let's say we start from the beginning, we've got these two blue lines to represent the original DNA double helix. So step one is the DNA will have to unwind and unzip. So therefore the enzyme helicase will come in and start to unwind and unzip the DNA all the way until here already. And from this point onwards, it will still continue to move forward to unwind and unzip this DNA right until the end of this DNA double helix. So now at this point, at this time, this part of the DNA already unwind and unzip. So all the bases are already exposed and ready for the new nucleotide to come in and join with the bases by complementary base pairing. Okay, so now you can see that after the helicase unwind, unzip, as it continues to move forward, the next enzyme will come in. So the second enzyme that will come in is the DNA polymerase. So DNA polymerase will sit in here and it will start to add in the nucleotide, okay, by complementary base pairing with the template nucleotides. So you add the nucleotide one by one, and you know that the nucleotide can only be added in a way that the chain will only grow one direction, which is 5' to 3'. So imagine this nucleotide is added in, the next one added in, next one added in. So it's added in one by one like this all the way in this direction because of enzyme specificity. So because of that, you can see that this strand will continue to grow, okay, as the DNA polymerase continue to move forward to add the nucleotide all the way. So this is continuously growing. So therefore, for this particular strand here, so the DNA replication in this strand is considered as continuous replication, right? Because this new polynucleotide strand grow continuously without stop, okay? So the process of DNA replication here is called the continuous replication, okay? And the new chain that is formed is called the leading strand, okay? The leading strand. Why is it called leading strand? Because it always moves forward, okay? It always moves forward because the 5' to 3' direction is always forward, okay? So therefore, this is called the leading strand. Because you know the leader is the one that always move forward, right? So therefore this is leading strand, right? Always move forward. Now what about the other template? Okay, we're going to look at the other template now. Now in the other template, same thing. After unwind and unzipping up to here, the bases of this template are exposed. So next is the DNA polymerase will come in, okay? But it will not come in at the beginning here. It will come in, let's say, for example, at a particular time, DNA unzip until this point. Okay, it unzip until this point. Then a DNA polymerase will come and sit in here. Cannot see, okay, can see, cannot see the color. Okay, I put darker, so you can see. So DNA polymerase will sit in here, can see, and then it will start to add the free DNA nucleotide by complementary base pairing. So add free DNA nucleotide, okay, by complementary base pairing. So 2 marks already here. Add free DNA nucleotide, 1 mark, by complementary base pairing, 1 mark. Okay. So, and it must allow the chain to grow from 5' to 3' direction only. So that's the only direction. So if you look at the template, you see this end here is 5'. So that means for this chain, this end here is 3'. So that means this starting point is here. This is the 5'. Okay. So 5' is here. So therefore, this DNA polymerase can only add the nucleotide in the backward direction. Backward. Why I say backward? Because the chain is growing this direction forward. The chain is unzipping forward this direction. So this new chain is growing backwards. All right, because the direction of 5' to 3' is actually backwards. And this is the only direction it can grow. Okay. So therefore, you see, when the DNA unzip until this point, the DNA polymerase will sit in and add new nucleotide towards this direction. Okay, so 1 fragment of DNA is formed here. 1 fragment of DNA. All right, and then when the DNA polymerase unzip until this point, DNA polymerase. When the DNA double helix unzip until this point, another DNA polymerase will sit in here and start to add the DNA nucleotide also from 5' to 3' direction, so backward again. Then when the DNA unzip until this point, another DNA polymerase will sit in here and start to add the nucleotide, so again from 5' to 3' direction. So therefore, in this DNA template here, so you can see that now there are 3 DNA fragments that are formed already. Okay, so the chain does not grow continuously in this template. So therefore, we say that the process of DNA replication for this template is considered as discontinuous replication, right? Versus in this template, we call this the continuous, right? Continuous replication, because you only got 1 chain that grow continuously. So over here, you've got many chains that are formed, many fragments of DNA that are formed. So it's discontinuous replication, okay? And this new strand is called the lagging strand, right? The lagging strand. Why lagging? Lagging means it's slower, okay? It's slower. It moves backwards instead of forward. So laggers are the ones that are slower, and they're left behind one, okay? So they're laggers. So these are called the lagging strand because they move backwards instead of forward, okay? So therefore, when you produce the 2 new polynucleotide strand, one of them is called the leading strand because it moves forward. One of them is called the lagging strand because it moves backwards. All right, so here is continuous replication in this template. Here is discontinuous replication in this template. Okay, Ken? So number 1, DNA unzips, unwind and unzip, catalyzed by DNA helicase. Number two, there's a continuous replication in one of the template. Okay. Number three, there's a discontinuous replication in another template. And then number four, Okazaki fragments. So you know that there are a lot of fragments that are formed, right? So you eventually have to join these fragments together. You need to join the sugar phosphate backbone of all these fragments together using the enzyme DNA ligase. Okay. So therefore, overall, this is the direction of DNA replication based on the direction of how DNA unzip. Okay, unzip in this direction. So the chain DNA replication will be in this direction. So this animation for you to actually visualize how actually continuous and discontinuous DNA replication looks like. Okay, so you got replication fork, which continues to extend forward, and the new chains continues to be formed in opposite direction. Okay, one is continuous replication, the other one discontinuous. So you've got the leading strand moving forward and lagging strand moving backwards. Okay, do you notice there's this green color thing that I didn't mention? You see not this green color thing? All right, so what is this green color thing? I'll mention afterwards, okay? There's something very important, but not in your syllabus yet, but it's good to know, okay? Right, so diagrammatically, when DNA replicates, so you can see that these are the original template, so the parental strands, and then the dashed line are the new strand, okay, the daughter strands. So as the helicase move forward, DNA polymerase will add the nucleotide forward, okay, for the leading strand, backward for the lagging strand. So you can also use this diagram to show how the nucleotides are added in by complementary base pairing. Okay, so at the end of the day, once the entire DNA is being replicated, you will get two sister chromatids, right, held at the centromere region. So that means there are certain bonds that hold the sister chromatids together here. Okay, so can you visualize now how the sister chromatids are being formed by DNA replication? All right, so therefore DNA replication is considered as semi-conservative because the two new DNA molecules that are formed, each of them is made up of one old strand and one new strand, or one parental strand and one daughter strand. So that's the meaning of semi-conservative replication. So DNA replication leads to the formation of two sister chromatids. So this is another very good diagram that I find that you can also read it to understand the process, okay? Number one, helicase will unwind and unzip. Now, unzip is a very important word. If you don't write, you don't get marked in exam. Okay, you don't unzip, then you cannot copy the DNA. So must unwind and unzip the parental double helix. So pay attention to the word that they use to describe. It's very crystal clear words on. Okay. Number two, there is a continuous replication in one of the template. So the leading strand is the one that produce, right? So the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. So it's very, very crystal clear description. And this sentence itself, maybe you can get 2 marks or 3 marks because leading strand is 1 mark. Okay. Continuously, maybe it's 1 mark in 5' to 3' direction. And then DNA polymerase 1 mark. So one sentence, 3 marks. You see or not? So this is called good answer. Okay. Good answer. One short sentence, you can score all the 3 marks. So this is called good answer. Okay. This continuous replication occurs in another template. So what happened? The lagging strand is synthesized discontinuously. Now, primase, you have not learned, so let's ignore primases. So extended by DNA polymerase to form an Okazaki fragment. So now I want to introduce to you those fragments that are formed, right? The DNA fragments, they are called the Okazaki fragment. Okay, Okazaki fragment. So overall DNA replication direction is to the left side. Okay, to the left side. And remember, all the DNA are labeled 5' to 3', 5' to 3', all are being labeled, so it can be seen clearly. So there's one more thing here that you have not learned, I will mention, which is actually the primer. Okay, can you see this pink color structure here? And it's labeled as DNA primer. Okay, so why is there a DNA primer here? Now, because of the specificity of the enzyme, where's my enzyme? This enzyme, okay? DNA polymerase enzyme is very specific. DNA polymerase enzyme. This enzyme only allows its substrate to sit in, then only it can catalyze a reaction. And the substrate of this DNA polymerase, it must always be, remember, it must be a terminal DNA, terminal nucleotide, plus a free DNA nucleotide. Okay, so which means to say that, can this DNA polymerase join two free DNA nucleotides together? Should I call your name? Paywan?
Yes.
Okay. Based on your understanding of the substrates of DNA polymerase here, can the DNA polymerase join two free DNA nucleotides together? And Paywan?
No.
No, cannot, isn't it? Because the free DNA nucleotide is not the substrate that can sit in here. Can you see or not? It must be a terminal nucleotide that can sit in here. So free nucleotide cannot sit in. So which means to say that at the beginning of DNA replication, if this is a DNA template, this enzyme cannot join one free nucleotide to another free nucleotide. It cannot join one. Okay, so therefore DNA replication cannot start. So it can only start when there's a short chain of DNA here, a short chain of nucleotides here. That means at least maybe 3 or 4 nucleotides. So therefore, we need another enzyme, okay, to create this small chain of polynucleotide strand. Okay, so another enzyme must create this small chain of polynucleotide strand before this DNA polymerase can come in and then hold the terminal nucleotide to add another free nucleotide in here. Okay, so therefore, this red color short chain of polynucleotide is actually called the primer. And usually this is an RNA. So it uses RNA nucleotide. So we call it RNA primer. Okay. So that's why just now when you see the primer, these are all the RNA primer that must be added in first by another enzyme before the DNA polymerase can start to add in the nucleotide. So therefore, after the whole DNA process finish, there is another enzyme will come and remove this primer, okay, and then add in DNA nucleotide like this. And then the entire process of DNA replication is complete already. Okay, so therefore we move forward to see, okay, what we have. Look at the diagram just now. Okay, so these are the red color one are the RNA primer. So you see, you have to have this primer before you can extend the chain, right? You need the primer before you extend the chain. Now, how come the primer is not here? Because the primer already replaced by another enzyme. Okay, it's removed already and replaced with the DNA. Okay, so here also originally there must be a primer. So this is where I ask you, how come there's this green color? Can you see not? So the green color is actually the primer. Can you see? So if the green color disappear means there's another enzyme that remove it and replace it with the DNA nucleotide. Right? Okay, so this is a primer. So you see number 4, after the RNA primer is replaced by DNA by another enzyme, which is also called DNA polymerase, okay, DNA ligase will join the Okazaki fragment to the growing strand. So DNA ligase will join the DNA fragments together. Okay. Okay, so now let us recap the steps involved in DNA replication. Step 1, the DNA have to unzip. Step 2, DNA replications occur where the new polynucleotide strands are being added in 5' to 3' direction. So continuous replication in the template where the template starts with 3' end. Okay, so when the template starts with 3' end, right, your new DNA strand will grow continuously because this is 5' for this new DNA. Okay, this is the template. So this one becomes the leading strand. So because always move forward. That's my primer. Prime. Okay. So this is continuous replication. Then we also have got another template where this continuous replication occur because this template starts with a 5' end. Okay, and of course the other end is 3' lah. So because new DNA strand can only grow in 5' to 3', so it can only grow this direction. So backwards, always backwards. So therefore, this is called the lagging strand. Okay, and this is the template. So the DNA replication is discontinuous replication. So Okazaki fragments are formed and they are joined by DNA ligase here. So they will be joined by DNA ligase. When this one has come close to it already, they can be joined by DNA ligase. Okay. So let's answer this question, past year question. Explain why cells that are produced as a result of mitosis are genetically identical. 3 marks. Why is it cells that produced by mitosis are genetically identical? 3 marks. So you must start from DNA replication first. Okay. So we start from DNA replication first. So number 1, DNA replicates semi-conservatively. So semi-conservatively means the original parental strand will act as a template for the new strands to be formed by complementary base pairing. Or if you want to describe, you have to describe fully. A pair with T and G pair with C, C pair with G. Okay, so DNA replicates semi-conservatively where the 2 strands will act as a template for the new strands to be formed by complementary base pairing. Okay, so idea that each strand acts as a template. So you give, always give the term, the biological term that describes something. So because you write the term, you get 1 mark. After you write the term, you elaborate. What does it mean by conservative replication? So you get 1 mark here. That means each of the strand act as a template for the new strand to be formed by complementary base pairing. So this is elaborating semi-conservative replication. Okay. So, and then because of this, the 2 double helixes produced are identical, or the these 2 new DNA molecules produced, okay, 2 DNA molecules produced are identical. So because of this DNA replication, they are identical. Okay, and then you also elaborate what do you mean by identical? Okay, and then after that, you need to describe how come the daughter cells have got identical, because these 2, which were sister chromatids, they separate during anaphase to the 2 daughter cells. So idea that the identical sister chromatids that are produced by DNA replication separate in anaphase so that each daughter cell will receive one. So therefore, the daughter cells are genetically identical. And the last marking point is if you describe the meaning of genetically identical means they have the same number and same kind means I teach you same type. Okay, same type. Okay, remember what is same type? Same karyotype. Karyotype number. You know, all the chromosomes in our cells are, they are numbered according to their length. The longest one is number 1. Then second longest is number 2, all the way until number 23, 24, 25, 25. Okay, sorry. What is it? 23. Okay, 22. Okay. Number 1 until number 22. Okay, are autosome. And then you got 2. Number 23rd is actually 23rd. Is a sex chromosome. Okay, better write 22 pairs of autosome and 22 pairs of autosome and 1 pair of sex chromosome. Chromosome. Okay. So 22 pairs of autosome. Why 22 pairs? Because these pairs have the same number of karyotype because same length. 1 pair of sex chromosome. Okay. Right. So with that, we have already completed the lesson for today, which is about DNA replication.